Hi students, this is Biswajit, your zoology educator. In this class, we are gonna complete the chapter digestion and absorption in one short manner. So basically, we are gonna study the story of digestion's physiology to rock in biology. So let's start. Okay, so when we think about digestion, the first thing that will come into our mind, that is nutrition. So what is nutrition? Nutrition is the process of obtaining food, and we are animals and our mode of nutrition is holozoic nutrition. We intake whole food into our digestive system, and there will be extracellular digestion inside our gut. Okay, now the food, what is food? For nutrients we need to take food, we need to ingest food. Food are composed of biomacromolecules and some inorganic micromolecules. Okay, like biomacromolecules. Who are biomacromolecules? Protein, fats, and carbohydrates. Protein, fats and carbohydrates, they are biomacromolecule, and every day's food we need to take them in a very higher amount. Okay, whereas for vitamins, minerals, we are taking a very little amount. And we need food for energy, to gain energy. It will boost our immunity, and it will help us for growth and repair. Okay, suppose I am 30-year-old and in my past 30 years I have consumed thousands of kgs of food, right? Some of the food has provided me energy, some of the food helped me to boost my immunity, some of the food has helped me to grow myself in my mass and in my cell number. Okay, now what is digestion? What is digestion? The process of conversion of complex food substances into simple substances which is diffusible through our body cells. And here one mechanical or biomechanical machine is helping us, that is digestive system. Means whatever food we are taking, they are macromolecules, and they are polymers. They are polymers, long chain, like for protein, long chain of amino acids. Now this is the duty of our digestive system, to break down these units of the protein and give us amino acids, so that cell will become happier when they will see amino acids and they will start intaking those nutrients. Now digestion is accomplished into ingestion, digestion, absorption, assimilation and egestion. Okay, nutrition is accomplished into these 5 stages, and digestion is the second stage. Now the digestion going inside our body, that is of 2 types: mechanical digestion and biochemical digestion. What is mechanical digestion? Chewing of food, mastication, mechanical digestion. Churning of food inside our stomach, okay, to mix the food with the digestive juices, mechanical digestion. And obviously you know, peristaltic movement or peristalsis, kind of involuntary movement mainly, which will be shown by our esophagus, our intestine. So all these 3 are kind of mechanical digestion. Now biochemical process. Now when mechanically food will be grinded into paste, now it will be easier for the enzymes, for the catalyst, to break them down into simpler form. So biological process includes some hydrolysis. With the help of water, we have some hydrolytic enzymes. They will be catalyzing the reaction of breaking down complex food material into simple one, and give us glucose, amino acid, fatty acid, glycerol, fructose, all these things. Now here 3 very important things written there. Bolus. What is bolus? After chewing the food in our mouth, in our buccal cavity, the masticated food which is mixed with saliva, and that masticated food mixed with saliva which is passing to the stomach, that is called bolus. Now what is chyme? Partially digested food from stomach which is passing into the duodenum, small intestine, that is chyme. That is churned food that is in stomach mixed with gastric juice which is acidic in nature because of hydrochloric acid. Now what is chyle? Chyle means the undigested food when digestion is completed into small intestine, then that undigested food will be passed to the large intestine as chyle. So it's the undigested food entering into the large intestine, and the pH of that food is alkaline. Okay, now let's see some other slide. Okay, so basically in this unit, one thing I have forgotten. This unit in NCERT they have started with this great scientist Alfonso Corti. Alfonso Corti. Why his renown? In the beginning of his life, he was an Italian anatomist. What is anatomy? When we talk about the internal structure of our body and organs, that is called anatomy. So, in the beginning of his career, he was cardiovascular. He was a researcher of cardiovascular system of reptiles. But later on, he shifted his interest into mammalian auditory system. Now, when we will study the ear, then we will come to know inside our internal ear, inner ear, we have a cochlea. We have a cochlea, and inside that cochlea there is a membrane called basilar membrane, and on the basilar membrane there is an organ which is responsible for auditory responses or auditory receiver, acting as an auditory receiver, that is organ of Corti. See, this organ of Corti named after Alfonso Corti. Now in this organ of Corti, it will have some hair cell. These are modified columnar cells. Now these hair cells can sense sound vibration and can convert it into nerve impulse. So basically for the discovery or for the research on organ of Corti, this Alfonso Corti, he is regarded. Okay, so our digestive system is made up of two parts: alimentary canal, the long pipe, the 9-meter long pipe from mouth to anus, and side by side we have some glands. Some glands are there which are not directly part of that pipeline, but they are providing this pipeline with some juices which are helpful in the process of digestion. They are called associated glands. Now, who are the associated glands? Salivary glands. 3 pairs salivary glands are there. Okay, liver and gallbladder. They will be donating, liver will be producing bile juice, and gallbladder will be storing and concentrating the bile juice, and ultimately it will be delivering it to the duodenum. Pancreas will be producing and delivering pancreatic juice. Now, alimentary canal is having few parts called mouth, buccal cavity, pharynx, the junction of our respiratory tract and digestive tract, esophagus, the food pipe, stomach, where food is stored for 4 to 5 hours and partial digestion of protein will take place, and intestines, large intestine, small intestine and large intestine, and ultimately the last part is anus through anal canal. Okay, so when we talk about this buccal cavity, again this buccal cavity is having 2 parts: buccal vestibule, the narrow chamber in between our lips and gums, that is called buccal vestibule. Oral cavity, inside the gum up to the pharynx, the portion is oral cavity, and oral cavity is having palate. The upper floor of oral cavity is called palate. Tongue, the taste receiver, it's also helping in mastication, is helping in pronunciation, talking, and teeth. Obviously, without teeth, mastication is not possible. Now intestine, there are two types, small intestine and large intestine. Though it's named small, but it's the longest part of alimentary canal, 6 to 7 meter long, whereas large intestine is only 1.5 to 1.7 meter long. But this large intestine is wider. Its average width of the large intestine is 6 centimeter, whereas this average width of small intestine is 2 to 3 centimeter. So large intestine is wider than small intestine. Small intestine is having three parts: DJI, duodenum, jejunum, ileum, and the large intestine is having three parts: cecum, colon, and rectum. So let's talk about this diagram. Okay, so these two are the lip, and the lip will be guarding our transverse slit. Transverse slit, the opening of our digestive system, that is mouth, and this transverse slit will be guarded by upper lip and lower lip. Upper labium and lower labium. Lips are also called labium. Now both the lips are attached centrally with our gums. Both the lips are attached. If this is the gum, upper gum, lower gum, teeth will be there. So both the leaves are attached with our gum with a thin flap of skin that is called frenulum, labial frenulum, superior labial frenulum and inferior labial frenulum. Now this is inside our mouth, buccal cavity. Now the beginning of the buccal cavity, this place, the space between our lips and gums, this place is called buccal vestibule, and inside that the whole cavity is called oral cavity. Oral cavity, and oral cavity is the opening of the oral cavity internally, that is called pharynx. And pharynx is a common junction of internal nostril, oral cavity, eustachian tube from middle ear, and ultimately one side of pharynx will be opened to the trachea through epiglottis. The initial part of trachea is called larynx, and backward to the trachea, that means the ventral side of our body, there will be our food pipe. There will be food pipe or esophagus. And the dorsal side, front side, there will be our windpipe. Now this esophagus will enter into the abdominal cavity, and this abdominal cavity is separated from thoracic cavity, chest cavity, diaphragm, through diaphragm. Okay, now at the center of diaphragm, it's having one hole called meatus. With meatus, through this meatus, this esophagus will enter into the stomach. Now the next part is stomach. Now stomach will meet with the U-shaped or C-shaped part of small intestine, that is duodenum. Now below diaphragm, both the side of stomach, we have some accessory glands. Like here, this is the liver, largest gland of our body. With liver, this green colored gland, that is gallbladder. Then below stomach, at this loop of duodenum, we have our pancreas. Okay, a leaf-shaped organ. Now the duodenum will mix with the next part of small intestine, that is called jejunum, DJ, duodenum, jejunum, and the N part, that is ileum. That is ileum. Ileum is the longest part of small intestine, and duodenum is the site of complete digestion, and the site of maximum absorption is jejunum. Each part is having different importance. Now, through a valve, ileocecal valve, this ileum will open into large intestine, and the portion where in the large intestine the ileum is getting opened, this is called cecum. This is called cecum, and after cecum is having colon, upwards colon or ascending colon, parallel colon or transverse colon, downwards colon or descending colon, and extension of S-shaped descending colon, that is sigmoid colon, and after that it has rectum, the storehouse of fecal matter, and after rectum there is a very tiny tube-like structure, 3 cm long, anal canal, and ultimately anus. So this was a basic diagrammatic representation of our digestive system. Now, in different places of our digestive system, we'll be getting some sphincter, some valves like ileocecal valves. Like at the beginning of our stomach, there will be cardiac sphincter. At the end of our stomach, there will be pyloric sphincter. Now, what is the basic difference between these sphincter and valves? See, sphincters are always circular muscles. Sphincter are circular muscles, whereas these valves are fibrous membranous flap. Fibrous membranous flap. Sphincter are ring-like structure. Valves are flap-like, door-like structure, and we know that door can be opened into a single direction. Okay, so that's why these valves are utilized for to stop the backflow. Once you have entered, you cannot go back. So backflow, it's stopping the backflow of the food material or any material. Now sphincter is responsible for controlling the entry and exit of material. When sphincter is contracted, it will squeeze it will like narrow down the lumen of pipe. So here more amount of food was entering. After the contraction of the sphincter, less amount of food will enter. So it can control the entry and exit of material. So this is the basic differences between sphincter and valves. Okay, so now let's come to buccal cavity. Buccal cavity. So you know that buccal cavity is having the initial part of the buccal cavity, which is guarded by lips, upper lip and lower lip or labium. Now the space between lip and gum, that is called vestibule, narrow space between lip and gum. Now the upper floor of oral cavity is called palate. Upper floor of oral cavity, or roof, rather you can say the roof of oral cavity. The palate is having two parts. Anteriorly is called hard palate and posteriorly soft palate. Now anterior palate, if you see the palate anteriorly, it's having some folds, and at the posterior side is smooth. So those folds of palate, they are called palatine rugae. These palatine rugae are very much developed in carnivorous animals, and it helps them to hold the food inside the mouth. Now posterior palate is having uvula and tonsils. If you open your mouth, then you can see near to the throat there is a bell-like structure. There is a bell-like structure that is called uvula. This is called uvula, and lateral side of the uvula is having some lymphoid glands called tonsils. Sometimes you will get infection in the tonsil, right? So they will get swell up and you will feel pain. When there will be pharyngitis or laryngitis, then these tonsils also get infected. Now tongue, where is present? Tongue is present at the oral floor. Oral floor. Palate is present at the oral roof. Tongue is present at the oral floor. So the tongue is a freely movable muscular organ attached to the oral floor. And how tongue is attached to the oral floor? By a thin skin, then that is again frenulum, and this frenulum is called frenulum lingui or lingual frenulum. Now tongue is attached, is a freely movable, right? So it's free at the anterior end, but it's attached with the hyoid bone, one U-shaped bone near to our throat. This is called hyoid bone. The tongue is attached to hyoid bone. Okay, at the posterior side. Now tongue is having some papillae. Tongue is having some papillae. Let's see. See. Tongue is having some papillae. What are papillae? Papillae are this granular structure present on the surface. They are called papillae. Now there are 4 type of papillae are present. Fungiform papillae. They are the papillae present on the anterolateral side. Anterolateral papillae. And these fungiform papillae are equipped with taste buds. Taste buds. Now the most numerous papillae spreaded everywhere in the tongue. They are called filiform papillae, and they are numerous, but they are devoid of taste bud. No taste bud. No taste bud on filiform papillae. Now at the posterior lateral side, some papillae are there. They are called foliate papillae, leaf-shaped papillae. Though they can carry the taste bud, but in human, these foliate papillae are vestigial. Vestigial means there is no function of these papillae. Now, at the posterior end, most posterior end of the oral part of tongue, this part of tongue is called oral part of tongue. We have 8 to 12 largest papillae. They are called circumvallate papillae. Circumvallate papillae are also equipped by taste bud. And behind the circumvallate papillae, one V-shaped ridge or groove is there, that is called sulcus terminalis. And behind that, this part of tongue is called pharyngeal tongue. Pharyngeal tongue. Here this thing you can see, this is the uvula, the part of a soft palate. And this pharyngeal tongue, again we will get some tonsils, lingual tonsils, lymph gland. These tonsils are what? Lymph glands. Now when we talk about tongue, we must have to talk about our teeth. Now what are the basic information of our teeth? We know that, okay, one more important thing about our tongue. What are the function of our tongue? Function of our tongue. Taste bud. Okay. Gustatory receptors are present. Gustatory receptors. On our tongue is helpful in the process of mastication, that is chewing of food, mastication or chewing and mixing the food with saliva. Tongue is called universal brush. Saliva will contain some antimicrobial content, and with tongue will be cleaning our mouth. That's why universal brush. And sometime when you will visit the doctor, doctor will check the color of your tongue. Doctor will check the color of your tongue. Okay, so the color of your tongue is a signal either you are healthy or not. When it will become bluish, it will give you a signal that there is some hypoxia inside your body. If it's become yellowish, then there may be jaundice into your body. So that's why the tongue is called mirror of disease. And two minor things about tongue. Tongue also can produce a very little amount of fat digesting enzyme, lipase, that is called lingual lipase. Okay, tongue is having two glands, one Ebner's gland and Nernst gland. Now teeth. Teeth, they will help us in mastication, cutting and chewing, grinding, and also holding the things. When we will eat, I can hold the thing with my teeth. Okay, so teeth can hold the things. Holding is also one function of our teeth. Cutting, chewing and grinding. So we have 4 different types of teeth. We can see here. In the beginning, each half of our jaw, we have 2 jaws. Upper jaw and lower jaw. Upper jaw is immovable, lower jaw is movable. Upper jaw is called maxilla, lower jaw is called mandible. Maxilla, mandible. Okay. So in the jaw socket, in the socket of jaw, what are sockets? Some cavities on a jawbone. These are called socket. So this root of teeth are embedded. So that's why our teeth are thecodont. Thecodont. Our teeth are thecodont. Root of teeth firmly embedded into the jaw socket. Jaw socket also can be called alveoli of jaw. Alveoli of a jaw. So our teeth thecodont. In our teeth heterodont. Why our teeth heterodont? Yes, because we have 4 different type of teeth. Incisor, scissor. Cutting, function is cutting and holding. Canine, knife, function of tearing and sharing the flesh. Canine is very much developed in vampires, you might have seen that, or in carnivorous animals. Premolars, you can assume with this structure of premolar with nut cracker. They are also helping in cracking and grinding. And ultimately the largest teeth of our jaws, that is molars. They are the pestle and mortar grinder teeth. Okay, so that's why this arrangement is called heterodont arrangement. And one very important thing about this molar teeth and few of the premolar teeth also, if you check it, the upper surface of the molar, there will be some cusp like structure, some elevated cusp like structure, and this is the exclusive feature of mammalian teeth. So this kind of cusp, if teeth are equipped with cusp, then this kind of teeth are called bunodont. Okay, bunodont. So our teeth is thecodont, our dentition is thecodont, heterodont, bunodont. Now again, we have some milk teeth. We have some in one stage of life. In the initial stage of life, the first 20 teeth we will get, they are called milk teeth or deciduous teeth or temporary teeth lacteal teeth. How many milk teeth are there? 20 milk teeth are there. This is the dental formula of milk teeth. How you will be calculating the dental formula? You have upper jaw maxilla, you have lower jaw mandible. Now you take only one half of the each jaw. You take only one half of the each jaw and count how many teeth are present in the upper jaw and lower jaw. So like this, milk teeth, formula of milk teeth is 2102 by 2102. Total 20 teeth are there. Okay, you have to multiply this with 2. Now why this 2 by 2? 2 incisor upside, 2 incisor downside. 2 incisor here, 2 incisor here. One canine upper half of the jaw, one canine lower half of the jaw. This 1 by 1. Now 0 by 0. In infants there is no premolar teeth. This 2 by 2, these two are the last 2 molar teeth. But in one very important thing, in case of infants, the socket of premolars are occupied by the first and second molar. Okay, and after the deciduation of this milk teeth, when these milk teeth will be shaded off, then next set of teeth will appear. We call them permanent teeth. Now for permanent teeth, every teeth will occupy their own socket. Okay, so here we can see we have seen the few teeth. These 20 teeth, they will appear twice in our lifetime. So teeth which are appearing twice in our lifetime, they are called diphyodont. Diphyodont. And how many are diphyodont? These 20 milk teeth are diphyodont. If somebody asks you formula of diphyodont, then 2102 by 2102. Then few teeth will appear only once in our lifespan. Only once. They are called monophyodont. How many monophyodont? Permanent. How many permanent teeth are there? 32. 32. So we have 12 monophyodont. Who are monophyodont? Who are monophyodont? All the premolars are monophyodont, and the third molar monophyodont. Rest all the teeth are diphyodont. Incisors diphyodont, canine diphyodont, first and second molar diphyodont. So that was the things about our teeth. Okay, one more thing about our teeth. Our teeth, the layers of our teeth. So jaw bones are lined by gingiva. Jaw bones are lined by gingiva or gum. Gingiva or gum. This is jaw bone. This is jaw socket. This is jaw socket. Now the outermost part, the crown of the teeth, is made up of enamel. More amount of inorganic salt like calcium phosphate will be there. Okay. Now below enamel there is a yellow colored layer. Below enamel there is a yellow colored layer that is called dentine. It is dentine. Enamel is white in color, dentine is yellow in color, and below dentine the central cavity of our tooth are filled with nerves, filled with some unspecialized cells, some stem cells and blood vessels. So this cavity is called pulp cavity filled with pulp, and inside the pulp there will be blood vessel and nerves will be there. That's why if you get dental caries or dental cavity, You feel the sensitivity in your dental pulp. Enamel producing cells are called ameloblast. Dentine producing cells are called odontoblast. Okay, now we'll move to the pharyngeal region. So this whole part is the pharynx. Upper part of the pharynx is called nasopharynx. Oral part of the pharynx is called oropharynx, and the last part of the pharynx is called laryngopharynx. Laryngopharynx. Now, while you will be drinking or eating something, it will enter into the food pipe through opening. The opening of food pipe is called gullet. Is called gullet. Okay. Now food pipe is present toward the ventral side of our body, and this is the windpipe or trachea that is present to the dorsal side of our body. Now there is a chance that food or water will enter into the larynx and then trachea, and it will block the trachea and we will die. So how it's protected? It's protected by a cartilaginous flap on the opening of the larynx, that is glottis, through epiglottis. Through epiglottis, a cartilaginous flap. A cartilaginous flap. Okay. Now here again, while you are drinking and eating, a chance is there food will go inside your nose. So to stop that, here this uvula, the bell-like structure of soft palate, uvula is there. Uvula will stop the flow of food into our nostril. So here you can see it summarized everything, the layers of teeth, enamel, dentine. Enamel will be produced by ameloblast, dentine will be produced by odontoblast. Pulp is having nerve, pulp is having blood vessels, pulp is having lymph vessels. Okay, there is no cusp in teeth. We are talking about cusp now. There is no cusp in incisor and canine teeth. Now pharynx serve as the common passage for food and air. Already we have discussed this. The esophagus, trachea and eustachian tube open into the pharynx. That also we have discussed. Swelling. Now after in our mouth we will be chewing the food. After chewing we will be degluting, swelling the food. This swallowing of the food is called swelling process is called deglutition. Swallowing process is deglutition. Here we have spoken about epiglottis. That epiglottis, you need to remember, cartilaginous flap, right, made up of elastic cartilage which prevents entry of the food into the windpipe. Opening of esophagus is called gullet. Opening of esophagus is called gullet. Then what is esophagus? If you want to define it, is a posteriorly passing through the neck, thorax, and through hiatus opening is entering. It's like checking inside the stomach through diaphragm. Okay, so posteriorly, posterior to whom? Posterior to windpipe. Now, while esophagus is joining the stomach, so the opening of esophagus and the starting portion of the stomach is having gastroesophageal sphincter or cardiac sphincter, which regulates the opening of esophagus into cardia of the stomach. Esophagus will be delivering bolus into the stomach, right, through peristalsis movement. Now when the bolus will enter into the initial part of stomach, that is called cardiac portion or cardia, that will be regulated by gastroesophageal. Gastro means it's related to stomach. Esophageal means one part towards the stomach, another part towards the esophagus. Gastroesophageal, or as because this is present at the beginning part of the stomach, cardia, also you can call it cardiac sphincter. Now about stomach. Stomach is the J-shaped bag which stores food for 4 to 5 years and is responsible for the partial digestion of food. Where it is present? It's present at the upper left side of our abdominal cavity. Upper left side, below diaphragm, upper left side of our abdominal cavity, our stomach is present. See, the opening of the stomach here is guarded through cardiac sphincter. The beginning part of the stomach is called cardiac portion or cardia. Actually, in noun, this is cardia. Okay. The head, like upper portion of the stomach, is called fundic region. Now, this is the bigger part of the stomach. This is called body of the stomach. Body of the stomach. Here, all the game will be played. The partial digestion of protein. And the last part of the stomach is the pyloric end, or this is the pyloric end or pylorus. Now this pyloric end will be guarded by another circular muscle, pyloric sphincter. Now in the pyloric region and body region, some irregular folds are present. They are called gastric rugae. They are called gastric rugae. They are called gastric rugae. And inside the fold, these irregular folds of gastric rugae, gastric glands are present. Gastric glands are present. Now, if you want elaboration of gastric gland, gastric glands are made up of different types of cells like argentaphin cells. Argentaphins, chief cell or peptic cell or zymogen cell, parietal cell or oxyntic cell, and mucus secreting mucosa, neck cell. Argentaphin cells are of three types. G cell, there will be these argentaphin cells are basically endocrine cells. They are hormone producing cells of gastric gland. G cells produce the hormone gastrin and ghrelin. Gastrin and ghrelin, they are called the hunger hormone. While the food is entering from through peristalsis movement from esophagus to stomach will start churning movement. At that churning movement, there is a kind of reflex that will stimulate these argentaphin cells to and this G cell will start producing gastrin, and the gastrin will act upon other cell. They will be releasing HCl. They will be releasing the protein digesting, the inactivated form of protein digesting enzymes. Now this chief cell, why these are chief cell? Because the function of stomach is partial digestion of protein. In stomach, protein will be converted into proteose, peptones and peptides. Okay. Now this chief cell or peptic cell, they will be producing pepsinogen, one zymogen or proenzyme. And in infants, they can produce prorenin. They can produce prorenin in infants. Parietal cells or oxyntic cell, similar thing. They will be producing HCl with a pH of 1.8. This HCl is responsible for the activation of this pepsinogen into pepsin, prorenin into renin. Again, this oxyntic cell will be producing intrinsic factor, Cassel intrinsic factor. This Cassel intrinsic factor are responsible for the absorption of vitamin B12 into our intestine. B12 is, or cobalamin is called extrinsic factor, as because when there will be active absorption of vitamin B12, without this intrinsic factor, they will not get permission of entry into our intestinal layers. Now mucosa neck cell, there they'll be releasing mucus, and these mucus are rich in alkaline protein mucin. So that was about stomach. Now stomach will enter into the C or U-shaped structure that is called duodenum, the first part of small intestine. Now let's talk about small intestine. Okay, now this small intestine is distinguished into first C-shaped part, duodenum, and duodenum is the site of complete digestion. Important point. Then jejunum, this is coiled part of small intestine. Maximum absorption will take place here. Maximum absorption will take place into jejunum. Then last part, ileum. This is highly coiled and the longest part. Okay, moderate or very good amount of absorption will take place. So what are the things you want to remember? Duodenum is the site of complete digestion. Jejunum is the site of absorption, maximum absorption of food. Ileum also site of maximum absorption, also site of absorption, not maximum absorption, but is highly coiled, means the longest part of small intestine. This is duodenum, this is jejunum, and this is ileum. And ileum will open into the cecum portion of large intestine with ileocecal valve, because when chyle is entering, the undigested food entering into the large intestine, there won't be backflow of this chyle, this semi-fermented or primary stool. Okay, so at large intestine, so if you see the inner lining of this duodenum, jejunum and ileum, some absorption unit will be there, and we'll call them villi. Okay, absorption finger-like projection into the inner lining of this structure. U-shaped villi will be present into the jejunum, and some V-shaped villi will be present into the ileum region. Okay. So large intestine is having three parts: cecum, colon, and rectum. Cecum will host some symbiotic microbes. Okay, basically some symbiotic bacilluses will be there. At the beginning of cecum, there is a short finger-like structure is there. That is called vermiform appendix. There is no function of this structure, but in our ancestor, it used to harbor some microorganism like Ruminococcus, who used to produce cellulase enzyme to digest cellulose or raw plant material. But in case of us, generally we take cooked food material. We don't take raw plant material, so cellulose digesting enzymes are absent into our body. So there is no function of this appendix region. Why vermiform? They like worm, vermis. In Latin language, vermis means worm. So very little worm kind of structure. Narrow finger-like tubular projection. Now colon is having three parts: ascending colon, transverse colon, descending colon, and sigmoid colon. Okay, now if you check the colon, three bands of longitudinal muscles are present. These three band of longitudinal muscles are called tinea coli. Okay, and hostra, absorption reabsorption unit of water. Hostra will be formed by the combination of this longitudinal muscle and some circular muscle. So each bag kind of structure is called hostra, and each hostra is made by longitudinal muscle and circular muscle and the spongy inner skin of large intestine. So that's why it's written tinea coli, 3 bands of longitudinal muscle present on the outer surface of large intestine. Hostra, water reabsorption chambers made up of these longitudinal and circular muscles. Rectum, temporary storage of stool or fecal matter. When it's filled, then it will kick out the edgest out or defecate the stool through anal canal, and anus is the portion through which ultimately elimination of feces will be done. Then come to the histology of our alimentary canal. Histology of our alimentary canal. Wall of our alimentary canal or gastrointestinal tract is having how many layers? 4 layers. 4 layers are there. Outermost layer, it is made up of squamous cells, flat cells, and in few cases it will have some cuboidal cells also. So this layer is called, outermost layer is called serosal layer. Serosal layer is made up of epithelial cells, squamous epithelium and cuboidal epithelium, mainly squamous epithelium, and this is a mesothelial layer or mesothelium, inner lining of our, this outer lining of our alimentary canal, actually inner lining of our coelom or body cavity, that is called mesothelium. Now below this serosal layer we have a 2-layered smooth muscle layer, called muscularis. Now this muscularis having 2 layers, I said. The outer layer is made up of longitudinal muscle and the inner layer is made up of circular muscle. You can remember with the term low key. Sometimes we will be getting confused, which is outer, which is inter. The muscularis externa is longitudinal muscle, muscularis interna is circular muscle. Low key, L for longitudinal. You know low key. I think you all are fan of Marvel, right? Now next, below muscularis layer we have a submucosal layer. Now what is this submucosa? Submucosal layer actually made up of areolar tissue, nerve and some lymph and blood vessels. Connective tissue will be there. Submucosa is the layer which is attaching the underlying mucosal layer and muscularis layer. So the below submucosa we have a layer which is full of epithelial cells called mucosal layer. They are mainly made up of either columnar epithelial or cuboidal epithelium, and here in this mucosal layer only there will be some irregular fold called rugi will be present, okay, in stomach, and finger like folds called villi will be present into our small intestine. They are mainly responsible for absorption and secretion and absorption. In different places this mucosa is having different function. Okay, now in somewhere you have heard these 2 terminologies. Plexus is network of nerves. So there are two types of plexus are there. One plexus is called Auerbach's plexus, which is present inside the muscularis layer at the junction of loci, at the junction of longitudinal and circular muscle. That is called Auerbach's plexus. I am not repeating it again because this is not given in NCERT. And there is one plexus, one nerve network which is present in submucosal layer. That is called Meissner plexus. Meissner plexus is present in submucosal layer, and Auerbach plexus or myenteric plexus. Myenteric means the plexus present in muscularis layer. One more trick to remember this plexus is inside muscle or back plexus. Outside muscle means submucosa, Meissner plexus. So if you are starting, if you are forgetting the names also, if the plexus name is starting with M, that must not be present into the muscularis layer, because it's muscularis layer or back starting with A. Already we have discussed this thing. Mucosal layer is having some irregular fold called rugi, finger-like folds called villi, and the villi are lined by some brush border cells, columnar cells brush border, that is called enterocytes, with some microvilli. Brush border cells means cells with microvilli. To further, villis are finger-like projection, they are increasing the surface area of absorption. Further, these microvillis are increasing the surface areas in many folds. Gastric gland in mucosal layer of stomach, crypts of liver cone in mucosal layer of duodenum, goblet cell and mucosa neck cell in mucosa layer of stomach and intestine, their mucus secreting cells. Now in submucosa, this thing you need to remember, in duodenum, Brunner's gland are present into submucosa, will be releasing alkaline mucus and in small intestine, and this alkaline secretion of Brunner's gland, it will take part in formation of succus entericus also. Again in mucosal layer of ileum, remember this, there are some patches of lymph, there are some patches of lymph glands are present. Okay, they are called Peyer's patch. They are called Peyer's patch. Okay, one very conceptual thing I am saying, while we are getting OPV, oral polio vaccine, so why we are drinking the polio vaccine, they are taking it in the form of drops, why they are not giving injection? Okay, as because the polio virus will enter through mainly ileum region of our small intestine. Okay, there is the entry, that is the portal of polio virus entry. Now, while we are taking OPV, oral polio vaccine, they are having some dead or hit-killed polio virus. When they will try to enter through the ileum region, it's having what? It's having Peyer's patch, lymphoid glands. Now, those lymphoid glands will have B cells and T cells. They will come in contact with dead polio virus and they will get activated. Our immune system will get activated and our immune system will start producing anti-polio antibodies, anti-poliovirus antibodies. So whenever, if really we are getting infection through poliovirus, then our systems are ready. They cannot enter through the ileum. Now we'll talk about different glands and their secretion. Very first gland, we'll talk about salivary gland. Where salivary glands are present? Salivary glands are present into our facial region. How many salivary glands are there? 3 pairs. Below the ear or in cheek region, parotid glands, the largest salivary gland. Below the tongue, sublingual gland, and in between two jaws, between mandible and maximal, either submandibular gland or submaxillary gland. Okay, you don't have to remember the duck's name of salivary glands. Salivary glands will release saliva which are rich in water. 99.5% is water. 99.5% is water. Then it's having an antimicrobial enzyme lysozyme. Then tyalin or diastase or salivary alpha amylase. Similar names, synonymous names of this tyalin only. They are responsible for digestion of cooked starch or boiled starch. Then it's having some mineralic ions, sodium ion, potassium ion, chloride ion and bicarbonate ion. These 4 ions are present into our saliva, and our saliva is generally hypotonic in compared to our body fluid. Okay, and these four mineralic ions of the saliva condition these enzyme salivary amylase or ptyalin. Without these mineralic ions, saliva cannot adjust their pH. The pH of saliva is 6.8, slightly acidic, near to neutral. Now stomach will be producing mucus through mucosa nexal, prorenin from parietal cells, pepsinogen from prorenin and pepsinogen, these two are zymogen, will be produced by peptic cell. I was actually thinking about intrinsic factor. Intrinsic factor will be produced by parietal cells. Hydrochloric acid also produced by parietal cells or oxyntic cells, which is having a pH, HCl is having pH of 1.8, and gastrin produced by G cell, which is a local hormone. Already we have discussed in the stomach section. Now liver is having two anatomical lobe, left lobe and right lobe. Liver is having two anatomical lobe, left lobe and right lobe, and right lobe of the liver is little bit bigger than the left lobe. Okay, now these two lobes are connected by a ligament called falciform ligament. These two lobes of the liver are connected through falciform ligament. But internally liver is having many other lobes, like quadrate lobes are there, caudate lobes are there. But these two lobes, left and right lobe of the liver, is again made up of many polygonal structure. These are actually the functional and structural unit of liver, these polygons. These polygons of liver, they are called hepatic lobules. They are the functional unit of liver. These are made up of hepatocyte cells. Okay, there you can find this in hepatic lobules. You will be finding hepatocyte cells. You will be finding Kupffer cells, which are kind of phagocytic cells. This hepatic lobule will be producing bile or bile juice, and this bile juice will be stored into gallbladder. Now, what is the function of gallbladder? Function of gallbladder is to storage and concentration of bile. Okay, storage and concentration of bile. Liver is made up of mainly the type of cell that is columnar cells. Will form liver, the hepatocyte. Now this inside gallbladder, brush border columnar cells will be there in the inner lining of gallbladder. That's why they can absorb the excess water from the bile. So that is, they have one very major function, that is concentration of bile. Okay. Now there is a circuit of liver and the journey of bile that we have to remember. See, right hepatic duct will be produced from right lobe of the liver, and left lobe of the liver will produce left hepatic duct. Now both the right and left hepatic duct will join together to form a common hepatic duct. Okay, common hepatic duct. Now through the common hepatic duct, the bile will come down. Now this bile will take entry into the gallbladder through cystic duct. Cystic duct is the duct of gallbladder, and bile will get stored inside, and it will, after concentration and absorption of water, the gallbladder will release the bile again through the cystic duct into common bile duct. Now the common hepatic duct and cystic duct combinedly formed common bile duct. Okay, now common bile duct will come down further downwards. It will mix with pancreatic duct. The primary pancreatic duct and common bile duct will match again to form hepatopancreatic duct. Okay, now opening of this hepatopancreatic duct into duodenum will be there, where? Into the lumen of duodenum, with a bulged portion that is called ampulla, hepatopancreatic ampulla. Now these ampulla will be protected or guarded not protected, rather we say guarded. It guarded. This portion there will be releasing of bile juice, and that guarding will be by sphincter circular muscle, sphincter of Oddi. That is sphincter of Oddi. There is another sphincter, sphincter of Boyden, the common bile duct. When it joined with pancreatic duct, here also one sphincter will be there. It is sphincter of Boyden. So this tract you need to remember. Okay, I will explain it again with a diagram. Now bile. Now liver will produce bile, right? Liver will produce bile. What actually bile is? Bile is a mixture of water, some mineralic ions are there, the similar kind of ions, sodium ion, potassium ion, chloride ion, bicarbonate ion. Some organic salts will be there. What are the organic salts of bile? Sodium glycopolate and sodium tauropolate. Bile pigments will be there. What are bile pigments? Bilivardin. First bilivardin, and then bilivardin will be converted into yellowish pigment bilirubin. The pH of bile is 7.8. The pH of bile is 7.8. And also bile juice will contain cholesterol and lecithin, and this cholesterol and lecithin only will help in the formation of micelles or micelles. Okay. Now pancreas. Pancreas produces pancreatic juice as well as some hormones. So pancreas is a heterocrine gland. We know here we are studying digestive system. So the digestive juice, pancreatic juice releasing glands are called acini gland, acinar cells. There are some cuboidal cells are there. There are some cuboidal cells are there called acinar cells, and these acinar cells together will form pancreatic acini or acinar glands. They will be releasing pancreatic juice, and pancreatic juice have some alkaline salts like sodium carbonate, sodium bicarbonate, and some pancreatic enzymes. Pancreatic enzymes means they will be producing some pro-enzymes like trypsinogen will be produced. Okay, then chymo or trypsinogen will be produced. Pro-carboxypeptidase will be produced through the pancreas. Okay, so here I was talking about that circuit of bile. Okay, bile circuit. So here you can see this is the liver. This is the two lobes are there. Right lobe of liver and left lobe of liver, and they are connected by falciform ligament. Okay. Now this duct from right lobe is right hepatic duct from left lobe, left hepatic duct. They will combinedly form the common hepatic duct, and common hepatic duct with cystic duct. This is the gallbladder, right? This is GB, gallbladder. So the duct coming from gallbladder is cystic duct. So cystic duct and common hepatic duct joins together to form common bile duct. Common bile duct. Now this common bile duct will join again. This common bile duct will join with pancreatic duct. This is pancreas. So primary pancreatic duct will join with common bile duct, and common bile duct and pancreatic duct will form hepatopancreatic duct. Hepatopancreatic duct ultimately they will open into the duodenum. Okay, so that was the thing. See how amazing. This one slide is showing us that what is actually digestion. Proteins are getting punched through protease enzyme and producing amino acids. Okay, lock of lipids are broken by enzyme lipase, and we are getting fatty acid and glycerol. Starch are stubbed by amylase, and we are getting glucose. DNA and RNA, nucleic acids, are nailed by nuclease, and we are getting nucleotides, nucleosides, sugar, phosphate, nitrogenous bases. So this diagram actually showing us what is actually digestion. Okay. Now the physiology of digestion, how complex food will be broken down into simple food. Okay, so quickly we'll be completing this physiology. So first start with oral cavity. Our oral cavity will release saliva from salivary glands. Salivary duct will be fetching the saliva from salivary glands to oral cavity. Then oral cavity also have secretion of little amount of lingual lipase. So this is a negligible amount. Now this saliva is basically digesting the starch, and 30% starch is digested by salivary amylase. Means this, which starch? Why 30% starch? Only boiled starch or ripened starch or cooked starch are digested into our buccal oral cavity. Now this boiled starch will be broken by salivary amylase. See, this is starch molecule. So the molecules will be broken into pieces. Molecules will be broken into pieces and we will be getting disaccharide maltose. They are two glucose molecules connected by glycosidic bond, and that's why maltose is disaccharide. So function of salivary amylase or diastase or ptyalin is done. Okay. Now the bolus through esophagus peristalsis, movement of esophagus will take entry into the stomach. Now one very important thing here, just remember this, we know that taste buds are not only present on the tongue surface. Few taste buds can be present in the initial part of esophagus. In initial 1/3 part of esophagus is voluntary, then next 1/3 part of esophagus is having some voluntary muscles as well as some involuntary muscles. The last 1/3 part of esophagus is totally involuntary. Okay, now the bolus is entering through the esophagus into stomach. Stomach will be producing pepsinogen, stomach will be producing prorenin, stomach will be producing gastric lipase. Okay, now first we need to activate these pepsinogen and prorenin. For the activation of these two, HCl is needed. Acidity 1.8 pH is needed, and when HCl secretion will be there from the parietal cells, the gastric lipase will be destroyed. That's why there is no function or negligible function of gastric lipase. And we all know that in order to protect from HCl, stomach will be producing mucus. But one very important thing, the parietal cells, they are producing HCl. So you will be thinking that how they are not getting damaged by HCl. So the answer is parietal cells never produce HCl into in acidic form. They will be releasing H plus ion and chloride ion separately, okay, through the channels, through the protein channels. So when this H plus and chlorine ion will take entry into the lumen of stomach and they will get aqueous solution, then only it will start showing its acidity or acidic behavior. Okay, so HCl will activate protein. Here you can see the tertiary structure of protein. Protein will be broken by pepsin, one of the important endopeptidase. The pepsin will break down the protein peptide bond from inside, endopeptidase. Okay, with the help of HCl, and we will get peptones, partial broken pieces of protein. Okay, partial broken pieces of protein. We'll get peptones, we'll get proteose, we'll get peptides. Now done, protein. Now in case of infants, okay, casein, which is milk protein, casein, which is soluble protein in milk, that will be taken care by renin. Renin will break down this casein. Remember the spelling of renin. RENNIN. There is another renin, RENIN. That is a enzyme cum hormone released by juxtaglomerular cells of our juxtaglomerular apparatus of nephron. Okay, so don't confuse with that renin. That renin is actually indirectly responsible for increasing blood pressure. This renin is responsible for curdling of milk. They are converting casein enzyme into calcium paracasinate, which is insoluble format of casein, or basically they are catalyzing, they are breaking down, they are denaturing this casein protein, and when it will denatured into calcium paracasinate, we will get this curd. But renin is only secreted into infants, not in adults. In adults, the curdling of milk is taken care by our hydrochloric acid only. Okay, now chyme, after stomach that food will be converted into chyme. Now chyme has entered into the duodenum. Chyme has entered into the duodenum. Now while the chyme will enter into the duodenum, now see, the angry stomach is sending the chyme into duodenum through the regulation will be done the pyloric sphincter. And we know why stomach is angry, because stomach is producing a lot of HCl with pH 1.8. Now why, when this acidic chyme is entering into the duodenum, see how the duodenum is in fear. Okay, it's crying, it will start crying, because if this acidic chyme will enter into duodenum, the inner lining gonna get destroyed by this HCl. Like stomach doesn't have mucus, that much amount of mucus secretion, though the duodenum also having the goblet cell to produce mucus, but not in that higher level. Okay, now even the duodenal enzymes cannot get activated in acidic environment, so it needs some help who are at least alkaline in nature. Now while this will start crying, while this duodenum will start crying now, so few few hormones will be released by the duodenum, like CCK, cholecystokinin, secretin, okay, pancreozymin, pancreozymin. Now these hormones together, they will send a message to the sleeping gallbladder and sleeping pancreas that, bro, your little brother is in danger, HCl is coming to destroy it, you please save them. So they will get activated, okay brother, okay bro, no worries, we are sending some backup. So they will start producing their juices, and bile excited by CCK, cholecystokinin, gallbladder will also get squeezed and will release bile juice into the common bile duct, ultimately the through hepatopancreatic duct. Pancreatic juice and bile juice will be delivered into the duodenum, and when this the alkalinity of the pancreatic juice is near about 8.4 to 8.6. Alkalinity of bile juice is near about 7.8. So they will take entry into the duodenum. Now duodenum is safe from HCl, because HCl will be neutralized now, and suddenly our duodenum is happy now. And most of the remaining part of the food will be completely digested into duodenum. There is one more thing that is happening. Duodenum will release some more enzymes like enterogastrone, gastric inhibitory peptide. So these kind of hormones will be taking some signal to the gastric glands of stomach, and they will tell the angry stomach, yes big bro, you have secreted enough amount of HCl, now you stop secreting HCl. So gastrin of stomach will be inhibited, and secretion of HCl will be inhibited by enterogastrone and gastric inhibitory peptides. So now when duodenum is safe, digestion will start. Okay, so first this HCl is neutralized by some action of bile salts like alkaline salts of pancreatic juice, sodium hydrogen carbonate, sodium bicarbonate. And next, unless fats are not emulsified, fat digesting enzyme lipase won't get activated. So again bile juice will help here. Fats are present in large fat globules. Okay, now this fat globules will be surrounded by bile salt, organic bile salts, sodium glycocolate and sodium taurocolate. Now this sodium glycocolate and taurocolate, they are amphipathic in nature. Generally fat is what? Fat is hydrophobic. but for the function of lipase, it's a hydrolytic enzyme. Okay, mixture with water is needed, but fat are very much hydrophobic in nature. They're lipophilic but hydrophobic, so they don't get mingled up with water. So here these things will be taken care of bile salts. Bile salt will emulsify. How they will emulsify? They will sit upon the outer surface of the fat globule, and one hand of these bile salts are lipophilic and another hand of the fat are lipophobic and hydrophilic. Now with the lipophobic but hydrophilic hand, they will catch water molecule. That's why they are amphipathic in nature. Okay, so with one hand they are catching lipid, with another hand they are catching water. So there will be a pulling and pushing will be in between the lipid and the water, mediated by the bile salts, sodium glycocolate, sodium taurocolate, and ultimately after some time, due to the force generated, one kind of force will be generated, and the fat globules will be broken into small fat droplets, and that is called emulsification of fat. Emulsification of fat, which will be mediated by bile salts. Mixing of fat droplets with water, that is called emulsification. Okay, now the next process will get initiated. Okay, now first of all, pancreatic enzymes will start working. Trypsinogen, protein digesting enzymes, proteases are there. Trypsinogen is switch off form of enzyme that will be activated by one duodenal enzyme called enterokinase with the help of calcium ion. Calcium ion will act as a cofactor of this enterokinase. Remember, enterokinase is secreted from duodenum. It is acting on the pancreatic proteases. Trypsinogen is activated into trypsin. Now when trypsin is activated, it will activate chymotrypsinogen into chymotrypsin. Okay, it's like you have few friends, and when you are waking up, you need some alarm. So that alarm is enterokinase. When you wake up, you are calling your other friends. You please wake up. Now it's morning. So trypsinogen will convert it into trypsin. Trypsin will convert chymotrypsinogen into chymotrypsin, and again trypsin can convert pro-carboxypeptidase into carboxypeptidase. Now these all are activated enzyme. They will act on the remaining part of the proteins which are coming from the stomach, on proteins, on peptones, on proteases. These are small chains, these are polypeptide chains. Now all these enzymes, trypsin, chymotrypsin, carboxypeptidase, they will they will start cleaving the peptide bond and will get dipeptides. The digestion of protein is still remaining. Dipeptide means two amino acid molecules bonded together by peptide bond. Okay, now pancreatic juice also have one amylolytic enzyme called amylase, pancreatic alpha amylase, that will break down the starch into disaccharides and other complex carbohydrates into disaccharides. Who are disaccharides? Disaccharides are like maltose, disaccharides are like sucrose, disaccharides are like lactose. Okay, now fat is also there. Fat will be broken by lipase. This emulsified fat will be broken by lipase, and this activation of lipase will be done by bile juice. Again, fat will be broken into diglycerides. Fats are present in the form of triglycerides. Triglycerides will be broken into diglycerides, and again this lipase will break down these diglycerides into monoglycerides. Next, pancreatic juice. Pancreatic juice also have some nuclease enzymes. Who are nuclease? Nuclease are DNA and RNA, nucleic acid digesting enzyme. So nuclease will convert nucleic acid into nucleotides, and few nucleotides into nucleosides. Function of pancreatic juice is done. Now, intestinal juice, the juice of intestine, the juice of duodenum, that is called succus entericus, which is having a pH 7.8, will complete the process of digestion. Now, from the digestion of protein end products were dipeptides. Here the dipeptides will be broken by dipeptidase enzyme present in succus entericus, that is called iriopsin. Primary dipeptidase. It will cleave the bond this between two amino acid peptide bond, and we will get two amino acids. Now amino acids are free. Free. Now last part. From the digestion of carbohydrate, what we got? Disaccharides. Now these disaccharides will be broken further by disaccharidases. Like maltose will be broken by maltase, lactose will be broken by lactase, sucrose will be broken by sucrase. Okay. Now when you will break maltose, you will get two glucose molecules, monosaccharides. When you will break lactose, you will get one glucose and one galactose. Remember, galactometer is the instrument which is used to measure the purity of milk. So when you break lactose is a carbohydrate present in milk, right? So lactose will be broken down into glucose and galactose, and ultimately sucrose, which is abundant in sugarcane juice. Sucrose will be broken by sucrase or invertase, and you will get glucose and fructose. One glucose, one fructose. Remember, sugarcane, we consider that as a fruit only, na? So that's why when you will break down the carbohydrate of sugarcane, sucrose, you will get one glucose. One glucose is common for everyone. Okay, and another thing is fructose here. So carbohydrate also digested. Next, the remaining fats, disaccharides and diglycerides and monoglycerides will be broken by intestinal lipase, and the end product of fat will get fatty acids and glycerols. Now here some nucleotides and nucleosides were there. Now these nucleotides will be broken by nucleotidase present in intestinal juice, succus entericus, and we will get nucleoside. We will get nucleoside plus phosphate. Now this nucleoside will be broken by nucleosidase, and we will get sugar, pentose sugars and nitrogenous bases. So here digestion or biochemical digestion is over. Our next topic is absorption. Now our next topic is absorption. This digested amino acid, fatty acid, glycerols, glucose, fructose, okay, and pentose sugar, they will be utilized by cell. But first our body needs to absorb, the cells need to absorb them before the assimilation. So now we'll talk about absorption. Okay, so very quickly we'll be completing absorption. I think this is the most important topic of this chapter, most complicated as well as most important topic of the chapter, absorption. So there are four places in our digestive system where absorption will take place. First, in mouth, absorption will take place. Drugs. When sometimes doctor will give you some tablet and doctor will tell you to keep the tablet below the tongue. That's because inside our mouth, buccal cavity, below the tongue, the absorption rate is much higher than other parts. Stomach, absorption of water, absorption of alcohol, and absorption of some simple sugars. If you are directly consuming glucose, so that will be absorbed by your stomach. Even alcohol, if you are consuming alcohol, within some time only you will be feeling like you will be feeling the punch of the alcohol, right? So that is the reason is absorbed through stomach. Now small intestine, the prime place of absorption, and jejunum is the prime. Maximum absorption will be there, and also in ileum optimum, meaning very good amount of absorption will be there. In duodenum, rare absorption will be there. All the things, glucose, galactose, fructose, amino acid, fatty acid, glycerols, all the vitamins, minerals will be absorbed by small intestine. But if there is some remaining part into the undigested food, some remaining water, some remaining vitamins and minerals, they will be reabsorbed by large intestine. Reabsorption will be there in large intestine. There will be reabsorption of water, reabsorption of minerals, reabsorption of some vitamins like vitamin B12, vitamin B1, which will be produced by Escherichia E. coli bacteria, Escherichia coli bacteria, which are symbiotic bacteria of our large intestine, along with some labs, lactobacillus bacteria. There is some bacillus subtilis, bacillus megatriums, and obviously few drugs will also get absorbed through large intestine. So according to NCRT, where drugs are getting absorbed? Drugs are getting absorbed in mouth, and drugs are getting absorbed in large intestine. But in actually, different drugs are having different portals of entry. So what is absorption? If you want to define absorption, passage of digested food through mucosa into blood or lymph. Okay, so here absorption means it's a two-step process. First absorption is this is the small intestine. So digested food are present in the lumen of the small intestine. First this will be getting absorbed into the enterocyte cell. First it will be getting absorbed into the enterocyte cells inside the mucosa layer of small intestine, and from there it will be getting absorbed either in blood or in lymph depending upon the particle size. Now modes of absorption, this you need to study very nicely. How absorption, like mainly you have to remember here, among these materials, glucose, amino acid, fructose, galactose, fatty acid, glycerols, which mode of cellular transportation they will follow. Okay, so we know that there are two modes of transportation: active transportation, where there is utilization of ATP is needed, and passive transportation, where there is no need of ATP. Passive transportation will be there from with concentration gradient. High concentration to particles will move from high concentration region to low concentration region. It is called passive transport. But in active transport, particles will move from low concentration region to high concentration. So totally opposite phenomena will be there. Okay, now active transport is again of two type: primary active transport and secondary active transport. Primary active transport, for transportation, intracellular transportation upon cell membrane. On cell membrane there are some transporter proteins are there. Now if we are doing active transport, obviously one thing is common: ATP. You have to spend ATP. Now if that transporter protein is made for transporting a particular thing, then that is called primary active transport. It's like that you have a car, and that car is for you only. If you want to go somewhere, you have to spend some money and you can go from one place to another place. So that car is primary vehicle for you. Now your friend has a car. This car is not your car, but yes, if you are giving some money for sharing the fuel cost, then your friend is allowing you to come with him or come with her into this car. So that is called secondary active transport. So primary active transport will be followed by amino acid, cations like sodium ion and calcium ion, and vitamin B12. They will follow primary active transport. Secondary active transport, generally glucose. In case of glucose, secondary active transport is very much common. There are some transporter proteins who will be transporting sodium ions. So along with this sodium transporter, along with two sodium ions, they can take one glucose ion. So those two sodium, one glucose secondary transport will be there. Basically those transporter proteins are transporting sodium ions, but along with two sodium ions, they can take one glucose ion in expense of ATP. in expense of ATP. Galactose will also follow secondary transportation, and amino acid will also follow secondary transportation. Now, who will be following passive transportation? There are different types of passive transportation are there: diffusion, facilitated diffusion, and osmosis. Now, what is diffusion? If simply particles are moving from high concentration to low concentration zone through a semi-permeable membrane, that is called diffusion, and that this particle should be solute particles. Okay, now when solvent particles or water particles will move from high concentration to low concentration, that is called osmosis. So osmosis is only reserved for water. Who will be following diffusion? Amino acids, they will follow diffusion. Glucose, they will follow diffusion. All the fat soluble vitamins, vitamin A, D, E and K, they will follow diffusion method, as because fats also they follow the normal, they follow like to enter into the mucosal layer, they will follow the normal diffusion mode, as because these are tagged with fat A, D, E, K, they will also take entry into the mucosal layer through diffusion mode. Now all the negative ions, Cl minus ions and HCO3 minus ion, they will also follow passive mode, that is diffusion. Now facilitated diffusion. What is facilitated diffusion? A cell membrane is there, and upon cell membrane one transporter protein is sitting, carrier protein is sitting. Now this carrier protein will transport the molecules freely. They don't need to utilize any kind of ATP. Okay, so sometime without expending any energy, they will pass the proteins, they will pass the materials from one place to another place, outside the cell to inside the cell, or inside the cell to outside the cell. So when normal diffusion process is going on, particles are moving from high concentration to low concentration, but that is facilitated by, means here particles are getting one extra advantage, particles are getting extra advantage of carrier protein. Okay, so that is going more faster with the help of a carrier protein. Normal diffusion process when it's going on, so that is advantageous, so we call it as facilitated diffusion. Who will follow facilitated diffusion? Fructose will follow facilitated diffusion, glucose will follow facilitated diffusion, galactose will follow facilitated diffusion, amino acid will also follow facilitated diffusion. Now you will ask that when there is already diffusion, why this facilitated diffusion, why this extra facility is needed? The answer is sometime suppose fructose cannot pass through the membrane with a normal diffusion, maybe due to its particle size, maybe due to its polarity. So that's why it just need a favor, from carrier protein who don't need any expense of ATP. So without utilizing ATP, it will be passing fructose from one place to another place. Okay, so this chart you need to remember in a very well manner. See, almost everywhere you can find amino acid. Amino acid can be transported through active mode and passive mode. When glucose also can be transported through active mode and passive mode. Only fructose following facilitated transportation is exclusive for fructose. Fructose don't have any other mode of transportation. Negative ions only have one option, diffusion. Positive ion only have one option, primary active transport. Rest amino acids, glucose will find everywhere. They have numerous way to get transported from one place to another place. Now there are different forms of glucose are there, right? L-glucose, D-glucose. So depending upon that, transportation of glucose will also be changed, and amino acids also having different polarities are there. Few are positively charged, few are neutral amino acid, few are negatively charged amino acid. So all this polarity, the charge of the particles will be vary as because different amino acids are there. So sometimes they will be following primary mode of transportation, sometimes they will be following diffusion mode, sometimes they will be following facilitated diffusion mode. Now the next thing and the important thing is absorption of fat. So first, before absorption, fats are needed to get emulsified. fats are needed to get broken down. So with the function of lipase enzyme and bile salts, fat globules are broken down into fat droplets. Fat droplets. Okay, and these fat droplets are surrounded by these fat droplets are these pink color things are these pink color dots are actually bile salts, sodium glycocolate, sodium taurocolate. They will be surrounding the fat droplets into a spherical manner with the help of cholesterol and lecithin, and this spherical structure we call micelles. This spherical structure we call micelles or myceles, is as per your pronunciation. Now these micelles are actually the delivery structure of these fat droplets. The micelles will be carrying the fat droplets near to the cell membrane, and they will enter, the fat droplets will enter into the cell through the process of simple diffusion. This series bile salts are not entering into the cell. This is an enterocyte, cell of inner lining of intestine. Cells present, brush border cells present in the villi, enterocyte. So only fat droplets have entered into the enterocyte cell. Okay, into the mucosal layers, cells of mucosal layer of small intestine, simply speaking. Now what will happen? They will be get covered by Golgi complex. Golgi body will take them inside, and we know that Golgi are responsible for the conformational changes. Now what Golgi will do? Again Golgi will start reassembling of these fatty acids, and inside Golgi they will form triglyceride. Again large fat globules will be formed, and this time these are coated by protein, and this protein coated large fat triglyceride globules are called chylomicrons. Chylomicrons are produced by the Golgi complex of cell. Okay. Now these chylomicrons will be kicked out of the cell by the process of exocytosis. The chylomicrons will be kicked out of the cell by the process of exocytosis. Now this chylomicron, they cannot enter into the blood capillaries, as because the fenestra holes are very much smaller. Okay, so they cannot enter into the blood capillaries, as because the holes of blood capillary, the fenestras of blood capillaries are very much smaller, and the chylomicrons are larger than that. So instead the chylomicrons will take entry inside the lymph vessels called lacteals, or lymph capillaries called lacteals, which are present below the villi, as because the gap of lacteals, the fenestra of lacteals are much bigger, and the chylomicron will take entry into the lacteal, but near to some other place, near to the subclavian vein and all, the lacteals again will pass the chylomicrons to the blood. The process of absorption is described in a detailed manner. These are the finger-like projection villi. And on the surface of villi, we will be finding some cells with microvilli. Cells will be like that. Okay, enterocyte cell. The first absorption will be there into the enterocyte, and from enterocyte they will be entering into the blood. Now in which blood the absorption is going on? Actually, you need to remember this: deoxygenated blood. Deoxygenated blood is absorbing the nutrients. Blood is coming through capillaries, blood is coming through arteries, and through capillaries they are delivering the oxygen to the internal part of the villi or intestine. Now oxygens are exhausted here, the blood inside the capillary will become deoxygenated blood, and now deoxygenated blood through the veins, through the hepatic portal system, they will be reaching towards the liver. So deoxygenated blood is carrying, or venous blood is carrying the nutrients. Venous blood is supplying the nutrients to the cell. Okay. Okay. So what will happen actually? This venous blood will be supplying the through the liver. So liver is a screener, liver is a filter. It will filter the blood and it will check whatever nutrients are there inside the blood. If that is good for our body, liver will pass through. If that is not good, liver will entrap that. Okay, so that's why liver is called detoxifier of our body, and that will be ultimately circulated through the other parts. And similarly, lacteals are also there. These are the lacteals. Fat chylomicrons will be taken by lacteals, and similarly the lacteals can also supply to the body, and near to the subclavian vein the lacteal will share these chylomicrons inside venous blood. Last part of the chapter, that is disorder of our digestive system and digestion. Quickly we will be studying this. Gastroenteritis. Gastroenteritis means wherever you will be finding a disease name ending with itis, that called inflammation. That is the inflammation of GI tract, gastrointestinal tract, by the infection of bacteria, by the infection of virus, by the infection of helminths. Next, jaundice, when your hepatocytes are damaged. Why hepatocytes are damaged? If hepatic ducts are blocked, if hepatic ducts are blocked, or the capillaries or the blood vessels of liver or hepar is not working properly, they are also damaged, they are also blocked, then jaundice will occur. Then ultimately bile pigment will be deposited into your on the surface of your skin, nails and eye. Where deposition of bile pigments will be there? Skin and eye. So that way they will become yellowish. Okay, because bile pigments are not get screened or unable to come into the urine or unable to come into the stool or fecal matter. Next, vomiting. What is vomiting? Vomiting is the ejection of stomach. Vomiting is the ejection of the content of stomach through a reverse peristalsis movement. Through a reverse peristalsis movement, which is controlled by vomit center of medulla oblongata. Now, why suddenly your stomach will eject out all the things? Because of reason is alkalosis. Reason is alkalosis. When too much secretion of HCl in our stomach will be there. So actually, when our stomach is secreting HCl, H plus and Cl minus ion, it will intake HCO3 minus ion, bicarbonate ion. So our blood will become alkaline. That is called alkalosis. Our blood will become alkaline. When our blood will become alkaline, the vomit center is a chemoreceptor center, chemosensitive center of medulla oblongata. It will send a reflex action to our stomach to eject out the things. Now, diarrhea. Diarrhea is the abnormal or frequent bowel movement. What is bowel movement? Movement of undigested food or movement of fecal matter inside our colon or intestine is called bowel movement. So why diarrhea will happen? Why frequent bowel movement will happen? Why loose motion will happen? Due to the reduced absorption of large intestine. Now next, constipation, opposite phenomena of diarrhea. Here feces are retained within the colon. Irregular bowel movement will be there, as because too much absorption will be there and fecal will get stuck, primary fecal will get stuck into the colon region, and so there will be pain. Now indigestion, very common problem with us, a feeling of fullness, you won't be feeling hungry. Why it's happening? Improper digestion causes inadequate or enzyme secretion. Inadequate enzyme secretion is there, then this indigestion will take place. Overeating, बहुत ज़्यादा ठूस ठूस के खा लिए, ठीक है, तब ये प्रॉब्लम होगा. एंजाइटी, स्ट्रेस and food poisoning. If you are eating too much spicy food, they will act as a poison and they will stop releasing enzymes. So all these conditions will lead to indigestion. And lastly, we'll be ending this chapter with PEM, protein energy malnutrition. These are malnutrition or deficiency disease of protein, and energy means carbohydrate. Okay, carbohydrates are the instant source of energy for us, right? So there is a disease called marasmus, there is another disease called kwashiorkor. Now what is the difference? Marasmus, you can see in a child, a newborn infant who is below less than 1 year old. In marasmus, there is coupled deficiency of protein and calorie. That newborn child, even his or her mother is unable to produce milk. For a newborn child, the main source of protein is mother milk. Now if famine is going on, if war is going on, if there is no supply of food, then this kind of thing will happen. So marasmus is a combined deficiency of protein and calorie. That child is not getting carbohydrates, that child is not getting protein. Okay, there will be thinning of the limbs, thinning of the body, and emaciation. Emaciation means muscle won't be formed, and if there is any muscle still in the body, they will be getting drained off. It is called emaciation. But in case of marasmus, no swelling or no blotting of stomach, no blotting into abdominal region will be seen. But in case of kwashiorkor, there is a disease, kwashiorkor. Now kwashiorkor will take place in the children who are in between 1 to 3 years old, little bit grown up. Here it's only protein deficiency. That child is getting enough amount of carbohydrate, carb rich diet is getting or she is getting, but no mother milk. So in that case, what will happen? Kwashiorkor affected child will still some cutaneous fat is there, subcutaneous, still some subcutaneous fat will be there into the body. Okay, it's because some of the carbohydrate will be converted into fat. Edema, dropsy will be there, swelling of abdomen, swelling of lower limbs. Still emaciation will be seen into kwashiorkor. Lower limbs will be swollen up, belly region, abdominal region will be swollen up, but there will be no such muscular tissue in the body. There is kwashiorkor. Here we'll see what is calorific value of carbohydrate, protein and fat. So when you take 1 gram of protein, carbohydrate and fat, and if you are burning them, oxidizing them, the energy will be released or produced by them, that is called their calorific value. What is calorie? If you want to increase the temperature of water, 1 gram of water by 1 degree Celsius, okay, at standard pressure, then that value is called 1 calorie. Okay, so what is gross calorific value and physiologic value or biologic value of proteins, carbohydrate and fat? When you take 1 gram of protein, carbohydrate and fat into a machine called bomb calorimeter, and you burn them with presence of saturated level of oxygen, full oxygen is there, then the amount of heat energy they are producing. So protein will produce protein will produce 5.65 kilocalorie energy. Carbohydrate will produce 4.1 kilocalorie energy, and fat will produce 9.45 kilocalorie energy. Outside the body, inside a metallic chamber where there is supply of oxygen is there, but when inside the body oxidation of protein, carbohydrate and fat will occur, then 1 gram of protein inside our body will produce 4 kilocalorie energy. 1 gram carbohydrate will also produce 4 kilocalorie energy, whereas 1 gram of fat will produce 9.0 kilocalorie of energy. So that is the physiologic value. So inside our body, why the value is little bit decreased? As because when we are intaking this protein, carbohydrate, fat, and this oxidation is going inside the cell, the presence of oxygen is not like atmospheric oxygen. That is why the reason they're decreased in calorific value while their oxidation is going inside our body.